Okay. Howdy, my name is Cassandra Fernandez and Alondra Curiel. We're gonna present our Microbial Culture Media Lab Report 3. Okay. Introduction. In this lab, a variety of media were used, including blood agar, CNA blood agar, esculin agar, mannitol salt agar, eosin, methylene blue, EMB agar, McConkey agar, Salmonella, Shigella, SS agar, each designed to test specific metabolic properties or growth patterns. Additional specialized media such as motility medium and dialkylate medium were applied to determine bacterial motility and oxygen requirements, respectively. The study also incorporated microbiological techniques such as motility testing, hemolysis determination, and aerotolerance testing using anaerobic chambers and glycolate broth. These methods provide insight into microbial physiologically and are essential for accurate identification. By integrating selective differential and enrichment medium with diagnostic assays, this lab demonstrates how microbial characteristics can be systematically identified. The combination of media responses, colony morphology, oxygen tolerance, and hemolytic activity forms the foundation of microbial classification and clinical diagnosis. Materials for cultures. Clostridium perfringens, E. facilis, E. coli, Salmonella typhimurium, Shigella flexneri, S. aureus, A. facilis, K. phenomena, Listeria monocytogenes, E. facilis, Mycobacteria smegmitis, S. epidermis, S. pathogens unknown 294 and unknown 114. Materials medium, blood agar medium, CNA blood agar, bio agar, mannitol salt agar, EMB agar, McCooney agar, Salmonella, Shigella agar, motility medium, T medium. Materials tools. We use an inoculating needle, an inoculating loop, a Bunsen burner, and a test tube rack. Procedure. Each agar plate was divided into 3 sections to its corresponding bacteria. The others were divided into 2 sections for our unknowns. For each plate, they were labeled unknown or the organism name. And then to initiate the lab, we flamed until red hot, transferring out our bacteria. For the agar plates, we use an inoculating loop. For the slants, we use a sterile loop in a zigzag manner. For the broths, we use a sterile loop. We gently swirled in that one, and then the deeps and motility for the medium portion. A sterile needle was used to stab the medium along the line in the middle. And then for the thioglycolate medium, the needle was used to inoculate. Meanwhile, we stayed close to the Bunsen burner.
Results. So in the first one, the blood agar hemolysis test on K pneumonia, which we see right here. It's shadowy but looks normal. So we interpreted this as gamma, no hemolysis, and because there's no change in the agar around it. and the S pyogen, which is right next to it, we didn't see any growth. This could either be to human error or might have burned like due to burning the culture, but further reading in the lab manual, it should have been beta. SRS, this one right here, it's faint shadow-like ring around it that we saw. There's likely two zones. It could be beta because of these two zones. And E facularis, there was no hemolysis present, so we interpret it as gamma. And for our unknowns in 114, we saw a clear ring. So it's beta complete lysis of red blood cells. And the unknown 249, we saw gamma no hemolysis. For the CNA agar and E. coli, we saw no growth. Um, it's the colistin and nalidixic acid inhibits the growth. In the S. auris, we see nice growth, so it's gram positive. And then for the S. pyrogen, we don't see growth, but we should have, so that could have been due to human error. In the unknowns, we see lots of growth in 114 on this side. And it's most likely gram positive. And for unknown 294, we see no growth, so it's either gram negative or it could have been human error. For the bioescalin agar and E. facilis on this side, we see growth with black precipitate. This is positive. This is a positive result. And S. auris on this side right here, we see some growth and that's a negative, but we see no black precipitate, so it's a negative result. And for the S. pyrogens at the bottom, we see some growth as well, but no black precipitate, so it's also a negative result. For the unknowns, on unknown 114, we see growth with black precipitate around it, so it's a positive result. And for unknown 294, we see lots of growth, as you can see, and it kind of like smeared into the other end, or the other side, sorry. There's lots of growth, but there's no blackness. There's some brownish precipitate to it, but we still interpret that as a negative result. For the mannitol agar on S. auris, we see growth and has a yellow medium or yellow precipitate around it, and this just means that it ferments mannitol. And S. epidermis on this side, we see growth but no fermentation, but we do see a pink red precipitate around it. And aspergillus, we don't see any growth. This could be because it's sensitive to salt. And for unknown 114 and 114, we see growth with a slight yellow tint. When it's in the light, this could be human error, or it does ferment in mannitol because of the light kind of tricking us. But the unknown 294 has no growth. In the McConkey agar plates, the E. coli, which is on this side, has some growth with a pinkish coloring to it. So we interpret that as lactose. It's a lactose fermenter. For S. typhimerin, we see growth with clearest colonies, and that just means it's a non-lactose fermenter. For SR is here at the bottom, we don't see growth, and that's because it's gram positive. For unknown 114, we see growth with a pinkish colonies growing, and that's a lactose fermenter, and we also see the same thing with 294. For the EMB agar plates, E. coli, we saw a dark colony that was like purplish on this side right here. It should have been green. This is wrong. This was due to the agar plate not being heated long enough. So this was human error. On this picture next to this should have been what the E. coli should have looked like. But our K pneumonia growth was kind of purplish, and that could mean it's a lactose fermenter if the EMB also didn't affect the outcome of these other two. The S. tymphanium, the growth was clear, so it's a non-lactose fermenter. And for our unknowns, 114 was dark purple, meaning it's a lactose fermenter, and same for unknown 294. A dark purplish growth means it's a lactose fermenter. For our SS agar plates, E. coli on this side had growth with pink colonies, and that just means it's gram-negative and it's a lactose fermenter. The S. flexneri on this side, it was a colorless growth, so we saw this as a non-lactose fermenter and it had no H2S production. And for S. tifminium, it also was a colorless growth in a way with some tint to it, but the tint just means it has H2S production. And for the unknowns here, 114, we see growth with pink colonies, means it's gram-negative and a lactose fermenter. Same goes for 294, pink colony growth, so meaning it's gram-negative and lactose fermenter. For the aero tolerance test for E. coli, these are the ones that were not in the chamber. For E. coli, we can see how it's like whitish for all of them mostly. And for the unknowns, it's also whitish, clearish, large growth. These are the ones in the chamber. We have see like kind of a pinkish tint on it. For the E. coli, we can see that it grew better aerobically. Also for the A. faculus. But they grow in all conditions. For the C. perfinogens, we see no growth aerobically, so it's only on air anaerobic. And for 114, for our unknowns 114 and 294, they grow in all conditions. For our thioglycolate mediums, E. coli, we see on this side, had growth and it's should have been top heavy. We see it more at the bottom, but it should have been a facultative anaerobe. Um, for RC perfinogens, we see growth only at the bottom right here. And that's the obligate anaerobe. Um, and then for A. faculus, we see growth throughout, but it should have been denser towards the top as well. And for unknown 114, we see growth more at the bottom. And then our unknown 294, we see growth throughout. For our motilities, we did not get pictures of them, but our results for those would have been some of them were kind of these were the ones that we poked straight through. Some of them had dispersed and basically ate up the agar around it. And some one or two did not.
Conclusion. The motility and air tolerance test provided valuable information for differentiating the bacterial cultures. The motility medium demonstrated that certain organisms exhibited active movement while others remained confined to the inoculation line, confirming non-motile characteristics. In the thioglycolate medium, most of the bacterial cultures showed growth concentrated at the bottom, indicating an anaerobic or oxygen-sensitive nature. In contrast, the two unknown bacterial samples displayed growth throughout the thioglycolate medium, but were denser near the top, suggesting they were facultative anaerobes that prefer oxygen but can also survive in its absence. Along with these results, the culture plates showed visible growth of colonies, with some expanding and dispersing across the plate, further illustrating differences in colony morphology and bacterial behavior. Overall, this lab was successful in proving bacterial growth and in demonstrating how motility, oxygen tolerance, and colony characteristics can be applied to distinguish between obligate anaerobes and facultative anaerobes and non-motile species. Thank you so much for hearing this out. I hope y'all enjoy. Have a nice rest of your day.